Ladies and gentlemen, I speak as a scientist whose responsibility it is to provide facts, not speculations first. The chairman asked me to speak on the topic of dreams and reality, and I'm going to concentrate primarily on reality. We are, I believe, on the train to developing strategies for preventing autoimmune disease. And I think that we can harvest the knowledge accumulated over the past 30 or 40 years to see where the opportunities reside and how we can progress. And what I'm going to suggest to you, ladies and gentlemen, is that the methods for prediction of autoimmune disease, in fact, are at hand. And that relatively shortly, we should have tools available to us to predict that the train of autoimmunity is underway and that we can begin to intervene earlier. And then I'm going to suggest at the end that the dream that still awaits us is to develop benign interventions. I'm going to use two disease examples of the many autoimmune diseases. I'm going to use autoimmune thyroiditis as one example and autoimmune myocarditis as the other. Those are the two diseases that my own group has studied primarily over the years, and I think are excellent examples because they are autoimmune diseases that are important. Thyroiditis is the most common autoimmune disease, especially in women, and is especially important in the later years when it may be an important component of frailty in later years. Myocarditis is the major cause of heart failure in young adults below the age of 40 and the major indication of cardiac transplantation as the only treatment we have for myocarditis and the subsequent heart failure. And these two diseases can be replicated probably better than almost any other autoimmune disease on the list by examples in animals, and we have in both cases defined models using well-characterized antigens or fragments of antigens, peptides, to produce both diseases in experimental animals such as mice, and we have spontaneous examples of both autoimmune disease. So these are the two most complete examples of autoimmune disease in my judgment. I'm going to use bits of work that was done in my own laboratory, not because it's by any means the only or even the major laboratory, it's just that it is easier for me to cite these examples and show how this accumulated knowledge will serve us in our goal of getting on the train of autoimmunity and getting on as early as possible. These diseases, like all autoimmune diseases, have a genetic predisposition. And that first became clear in studies of experimental thyroiditis in 1971, when we were able to show for the first time what one gene that's responsible for increasing susceptibility for autoimmune disease is. They are the genes of the major histocompatibility complex. demonstrated first with thyroiditis, but now can be demonstrated with every autoimmune disease has been seriously studied, and is one of the commonalities, striking commonalities, of all of the diverse autoimmune diseases. That first study in Science was followed rather shortly by a second study using a spontaneous model of thyroiditis in the chicken, showing again that this spontaneous disease, unlike the experimentally induced form of thyroiditis, also is related to the major histocompatibility antigen, or a major histocompatibility gene, I should say, of the major histocompatibility complex. And our laboratory and many others spent much of the decade of the 1980s working out the details of genetic predisposition, showing, for example, that the most important single gene encodes the recognition of the, in this case, thyroglobulin antigen or thyroglobulin peptide that initiates the autoimmune process, that gets the train going, if you will, showing that this by itself is modified by additional genetic traits within the IA complex. That by itself is of great interest, but also to show that non-MHC genes are roughly equal together, so that we have, in fact, a genetic predisposition balance between genes within the major histocompatibility complex and genes outside of the major histocompatibility complex that determine overall susceptibility. Those within the MHC2 genes are mostly involved in recognition of specific epitopes, specific antigenic determinants that provide susceptibility. Those outside are genes generally that regulate the immune response. All of these genes have different degrees of expression. That's something we now think of as epigenetics or post-genetics. And by far the most important elements determining expression of genes are the steroid hormones, the sex hormones, and by means of sex-transforming surgery and administration of steroid hormones, we could show that we could convert a susceptible female to a resistant male, or vice versa, by reversing the sex of the animal. So we don't change the genome of the animal, we change the expression of the genes by changing the hormone environment. We also emphasize the importance of both the induction of the immune response, however that may be, through molecular mimicry or through other means, but the critical role of adjuvants and the ability to induce disease experimentally depends as much on the appropriate adjuvant environment in which the antigen is provided to the animal as it is to the antigen itself. Those are equally important, and the importance of the adjuvant, I know, will be discussed by Professor Schoenfeld in a few minutes. In most occasions, the earliest marker that the train is underway, that autoimmunity is going on, not autoimmune disease, but autoimmunity is occurring, is the appearance of specific antibodies. And we have more and more come to see from this and other examples that antibodies can be used and are being used clinically as early predictors of autoimmune disease, not as diagnostics for autoimmune disease, but as predictors or as signs of autoimmune disease. And we've seen from examples in including thyroiditis, that some types of antibodies, for example, antibody to thyroid peroxidase, one of the major thyroid-specific antigens, are present only in relatively small amounts, but tend to rise as the patient progresses down the trainway to the appearance of clinical disease. Interestingly, the other major thyroid antigen, thyroglobulin, does not increase over time during the roughly decade that we were able to follow these serum samples, because, ladies and gentlemen, that antibody was present long before. That, in fact, is the first antibody that gets the train started very early. It is joined by a second antibody and a third antibody later in the course of events. So there is, I like to call it, an escalation of the autoimmune response. And that is an indication that we are proceeding from a subclinical, non-clinical autoimmunity to a clinical, pathological autoimmune disease. It is, in fact, evidenced by the appearance of additional autoantibodies. And we could show that in experiments using primates, which produce multiple autoantibodies, unlike mice, which produce only the single thyroglobulin antibody. Primates, rhesus in this case, produce antibodies to thyroid peroxidase. And while it is the response to thyroglobulin that starts the train going, it's the appearance of a second antibody to thyroid peroxidase, a completely different autoantigen, and also to the iodine SIM transporter, even a third antibody. It's the accumulation of several different antibodies that really is an indication that the train is accelerating and that we are going from a subclinical, actually harmless autoimmunity, but an increasingly severe one, because those are the antibodies that are evoked by an ongoing local adjuvanted autoimmune response. So that's the signal that we need to really be concerned with the pathologic outcome. That can be extended even more on a molecular basis by experiments that we've done looking at the immune response to the thyroglobulin molecule itself. And we could find that in those individuals who have thyroglobulin autoantibodies, which is very common, about 18% of us do, they develop antibodies to a restricted number of very conserved epitopes on the thyroglobulin molecule. When they transform from individuals with antibody to individuals with antibody and disease, that is hypothyroidism, then that's marked, that's signaled by the appearance of additional antibodies to other determinants, in this case more species-restricted determinants on the thyroglobulin molecule. That seems to be true not only for thyroglobulin, but now has been found with other autoantigens too. So it is in fact this antigen spread, as it's now known, which is an indication that, again, the train is moving faster and faster and faster as we accumulate more and more autoimmune responses, as signified by additional autoantibodies. And we can even use that for as a sign for the appearance of additional autoimmune diseases, because I think all of us realize that one of the characteristics of autoimmune diseases is that they tend to travel together. And an individual or a family with one autoimmune disease is somewhat more prone to a second or a third. And we can see that in this case, by the development of thyroglobulin antibodies in patients who develop another autoimmune disease, hypophysitis. So these antibodies can be cross-predicted also. Finally, we come to the train wreck. Finally, we come to the point where we cannot reverse the process, where we have to treat the symptoms, the result of the injury, in order to keep the patient alive. And in fact, that's where we are today. And we are quite successful in doing it. So patients who, when I started, were given a diagnosis of lupus and told they had 5 years to live, now can live a full lifespan in many, many cases, if carefully treated. But that means lifelong and very expensive treatment. So now we turn to myocarditis. And this is a disease which we can induce by a virus, Coxsackie virus, and we can follow the train of events from the genetic predisposition to the viral infection to the signals that tell us the progression of the disease from a self-limited viral infection that many of us have had and many of us have even had and never been aware of, to a pathologic and fatal outcome. So myocarditis begins, and this is now a mouse model that I think replicates what occurs in humans, with a viral myocarditis, which in most of us is an acute but limited disease, which rarely comes to clinical attention unless the patient is stressed or has already had some cardiac impairment. So in most mice and most people, that disease self-heals, it's self-limited. But in some individuals, this disease turns into a chronic form, and those individuals and those mice have genetic markers of that disease, but they also have cytokine markers of that disease. And those are increased production of certain key cytokines, and the two in this particular disease that are early markers of a later autoimmune myocarditis are interleukin-1 beta and TNF-alpha. Now those are common indicators of inflammation, and that's not terribly surprising. What was encouraging is to show that even in the most genetically susceptible mice, the ones that are most likely to develop this continuing and autoimmune myocarditis, we can prevent the disease by blocking either one of these two cytokines, especially IL-1, which is relatively easy to block at the receptor level. But the real evidence came by showing that we could induce the autoimmune disease in generally non-susceptible, genetically non-susceptible mice by giving recombinant IL-1 or TNF-alpha. So they are both necessary and sufficient during the first hours of infection, during the first 12 hours of infection. These two cytokines are changing the innate immune response to a virus to later induce, 9 days later, induce, at least begin, an autoimmune process. And that's, I think, a really key finding that suggests that it is possible to find early warning signals even before there is any pathologic, to say nothing of clinical evidence of disease. Now, in those animals that do develop the autoimmune disease, the severity of the disease is determined not by a single cytokine or even two cytokines. It's determined, in fact, by the cytokine profile. And I've shown on this slide just some of the cytokines that we have dissected out as being those that tend to facilitate, to promote the disease, and those that tend to retard it. And they are not necessarily classically divided into type 1 and type 2 or type 17. It is not a single cytokine. It is, in fact, the profile, the pattern of the cytokines that determines two important things. It determines the amount of inflammation, and that's important because the patients can die of autoimmune myocarditis, not usually, but they can. But it determines the nature, the quality of the inflammation. For example, in certain circumstances where IL-4 and IL-13 production are prominent, mice can develop the eosinophilic form of myocarditis. That is the one form which is the fatal form. It causes early death in young people. It's not common, but it is by far the most dangerous form. So it's both the amount, but equally important, the quality of the inflammation that determines the outcome of disease. And then we get to the last stage, the disease we call dilated cardiomyopathy. Now this is a disease in which we have fibrosis replacing the myocardial sites. Myocardial sites contract and let the heart carry out its physiologic function. Fibrotic cells don't contract, and when they are deposited in the heart as a result of inflammation, that, ladies and gentlemen, is part of the wound healing. That's what's supposed to happen elsewhere, and that's a good thing. But it's not good in the heart, because having non-contracting fibroblasts and collagen deposited in the heart means we have a large, flaccid, non-pumping heart. And these are the young people, these are the young adults, the 20 and 30-year-old, particularly males in this case, who die of heart failure and for whom we have really no effective treatment other than mechanical support temporarily and then cardiac transplantation. And here we have some early information, I think for the first time, of what the critical elements are in determining that an inflammatory process is underway. And interestingly, today's fashion in cytokines is IL-17. IL-17 is not a major player in the inflammatory stage of myocarditis, but it is essential for the development of dilated cardiomyopathy. So if we block it genetically, or if we use anti-IL-17 antibody early enough, meaning, I don't have a time scale here, about day 21 in the inflammatory process to about day 35. So in a critical period where inflammation is resolving, if we block IL-17, we can prevent the development of dilated cardiomyopathy, even in those mice that are genetically destined to develop this dire outcome. So that's why I think there's enormous hope. So we can dissect car by car, stage by stage, this train that gives us the pathologic outcome of the autoimmune response, autoimmune disease. We have, I think, at our disposal, if we're clever enough and hardworking enough to use this kind of basic information, we could develop within a reasonable period of time, I think probably half a decade, predictions for many autoimmune diseases. Our goal then is to intervene at some stage in this train before we reach the dilated cardiomyopathy, before the heart becomes fibrotic, because while we can treat and while we can even sometimes reverse inflammatory disease, we cannot treat, we cannot reverse fibrotic disease. So what do we need? What is the unrealized part of the dream that our chairman has challenged us with? Ladies and gentlemen, it's the benign intervention. We need an intelligent way, or ways of doing something for individuals who are not sick. And that's a big responsibility for any physician. We would be dealing with people who are not ill, who are not sick, and we are going to be intervening. We are going to be treating with something because we feel, based on the science available to us, that they are on the train to develop a train wreck. It's a big responsibility for any physician. It's a biggest responsibility if we're dealing with children. I believe we can do that, but I believe that's the task that is requiring the greatest amount of scientific engineering and ingenuity. So here's just one example of a benign, almost benign intervention. It happens to be, again, with myocarditis. And in this case, we know exactly what the antigen is that starts the process. Not the final antigen, because there are multiple antigens. But when myocarditis begins, it begins because there is an autoimmune response to a particular determinant on cardiac myosin, that's heart-specific myosin, heavy chain. And we have a 9 amino acid sequence, which is the peptide. That's the antigen, which is responsible for starting the autoimmune process about day 9 in our mice. If we alter that antigen, if we make an altered peptide ligand, which is what this LEAPS is, and administer it early in the process, we can prevent the ongoing disease and prevent dilated cardiomyopathy. And in fact, even if we give it during the inflammatory disease, we can reverse the disease to some extent. So I think I see great hope in doing this. This, however, is not something that could be translated as it is to humans because there are still complications with this kind of approach. But to me, it is a prototype that if we know enough about an autoimmune disease, if, for example, we know the precise determinant that starts the pathologic process, that starts this train to go off the tracks, then we can intervene. So this, to me, is a model of a benign intervention. There are others that we can think of, and there are at least two of them available right now. One, of course, is the treatment of streptococcus in children who are susceptible to rheumatic fever. Tremendous success in at least the industrialized countries, although not successful, mostly for economic and social reasons in the developing countries, especially in the Middle East. So there are other problems that go with that. Or in the case of celiac disease, again, a restricted diet, eliminating gluten from the diet, will prevent the development of celiac disease, even in genetically susceptible people. So we're not talking, ladies and gentlemen, about very expensive interventions. We are talking about things that don't require genetic engineering, things that could be done, but that require an understanding of the train of events that gives rise to pathogenic autoimmunity. So let me give you a summary of what I've said, and I've really said two things. I've said there are genetic and epigenetic data. Appearance and escalation of autoantibody, the autoantibody response and cytokine signals, these can be personalized and they can be predictive of an oncoming autoimmune disease. That is feasible. That is something within our grasp. Future application depends, in my opinion, upon developing benign interventions. And so I leave you with the same train of events that I described during the initiation of this. We are on the track. We are on the way. I think there are realities, Mr. Chairman, in this field. There are real discoveries. We really can utilize the information gathered over the last half century of diligent work in the laboratory and in the clinic, because most of these can be repeated in the clinic. But we have a lot of work to do, and I think we should get about it. We need benign interventions. Thank you very much.
You and we talk about mostly about pathological antibodies and antibodies predicting disease. So, to say bad guys. What about good guys? Antibodies predicting good course of disease, good course of beginning disease, and potentially antibodies with a potency to treat disease.
See, I'm audio-visually impaired here. Thank you very much. You're lots of help. You're asking about good autoantibodies. Yeah. How do you define it? What's an example of a good autoantibody?
I mean antibodies for prediction of a good course of disease.
Yeah. Yeah. You raise a question a chairman may want to comment on too, and that is, are you thinking of the evolutionary function of antibodies as natural autoantibodies? Is that the context that you're considering? Okay. Perhaps one of the most interesting ideas, which I think, by the way, would make Metchnikoff very happy, because Metchnikoff was a Darwinian and believed that most of what we have is of benefit. And really, he was really the first, I think, to propose the idea that antibodies have a function of benefit, and thought of them as the scavengers, the garbage collectors of the system. The evolutionary origin of antibodies were things to get rid of the waste products of cells. And that's something that one of my own teachers, Pierre Grabar, championed very much. I think you'll agree he was very enthusiastic about the transplanteur theory. It's certainly, and I think the concept for that, but the evidence for that, the concept is a strong one and makes to me a lot of sense. The evidence for it has unfortunately never been very good, because it's a very difficult thing to deal with. And it's difficult to deal with because it's very hard to know, to understand the process of cell death. We hear a lot about damage and injury of cells. We even have a danger theory of immunology, which I've always thought is a little bit funny, because it's hard to think the immune system is smart enough to know danger when it sees it. But the idea is that when cells die, sometimes that's perfectly normal and doesn't evoke a harmful autoimmune response, and sometimes it's not harmful, as normal apoptosis should be. So I think what we need to have is a much better understanding of what is good injury, harmless injury, what is harmful injury, and what is it that can evoke antibodies that are not dangerous antibodies, and antibodies that are more likely to sign danger. I think we would agree, by the way, that the antibody that we measure, at least, the antibodies we know about are usually not the dangerous antibodies. They're the accompanying antibodies. They're the signs of the ones we can measure because they remain nicely in the serum, and those are the ones we can look at. It's the ones that either don't remain in the serum for whatever reason, or are never produced, that are probably the really dangerous antibodies. And we still don't have an idea, at least I don't have an idea, I may be the only one left who doesn't understand how much of autoimmune disease is due to antibody and how much of it is due to non-antibody cell-mediated immunity. And until we understand that, I don't think I can give the full answer to the question, the very good question that you asked. Dr. Schoenfeld to translate this question to you.
Доктор Строев, Санкт-Петербург. Уважаемый профессор, спасибо за вашу лекцию. У нас в России, чтобы поставить диагноз иммунного тиреоидита, врачи ждут определенного титра антител к тиропероксидазе. Нам кажется, что иммунный тиреоидит начинается тогда, когда появляются уже единичные тела к тиропероксидазе, а вот антитела. Как вы смотрите на это? Так и прав я или нет?
Dear Dr. Rose, thank you for your brilliant lecture. The question of Dr. Stroyev from St. Petersburg regards to the clinical aspects of autoimmune thyroiditis. In Russia, clinical doctors commonly wait for certain minimal diagnostical titer of antiperoxidase autoantibodies. Do you think that clinical doctors shall wait for a certain level of autoantibodies, or the intervention must be started as early as possible with minimal level of antibodies discovered?
All right, there are several questions here. I don't know how much time I should take from your meeting. I can make a trade with Professor Schoenfeld and take some of his time to answer. So let me answer a couple of questions. All right, this won't be that long. I think one of the questions you were asking were the different kinds of antibodies. And in the case of thyroid disease, that's a very pertinent topic because I think we understand what the procedure is, and I referred to some of our early studies on rhesus monkeys, but now we have human data that seems to confirm that. Thyroiditis begins with an autoimmune response to certain epitopes on the thyroglobulin molecule. And that begins before 10 years, because we've gone back 10 years, and the thyroglobulin antibodies are still there, even before that period. However, those patients are euthyroid. They have normal thyroid function. We have no reason to treat patients who have normal thyroid function. We treat them endocrinologically. When that process begins and we begin to find inflammation going on in the thyroid gland, then we develop antibodies to other thyroid-specific, thyroid-limited antigens within the thyroid gland. And the most prominent one is thyroperoxidase. So that's the second antibody that occurs. And that can occur, and I think our slide showed that, although it was perhaps hard to read, 6 or 7 years before the patient is hypothyroid. The patient is normal, but we're beginning to see a rise in the antibody to thyroid peroxidase. So that's an early warning sign that we're moving from what I call benign autoimmunity to pathogenic autoimmunity. We don't treat those patients yet because we treat them endocrinologically. But I think that's why the antibody to peroxidase is clinically a more interesting antibody, although we prefer to look for both antibodies. Interesting enough, the thyroglobulin antibody can disappear and you can find only the peroxidase antibody in a certain proportion of patients. We don't know exactly why it disappears. We think it's because too much of the antigen is used up and there's not enough stimulus. Antibodies don't just go on and on. So that's I guess, the first question that you were asking about the sequence of antibodies. What we're also seeing, and maybe this gets a little bit to the second kind of question you were raising, it's not only the specificity of the antibody, it's the subclass of the antibody. So we're also finding that there is a change in an autoantibody subclass, and interestingly, a movement towards IgG4 in the case of thyroid disease, which indicates that we tend to be going from a fibrotic thyroiditis, from an inflammatory thyroiditis to a fibrotic thyroiditis. So that's the same specificity of antibody, but a different subclass, going from IgG2 antibody, which is the main one, to an IgG4 antibody, which is relatively uncommon, but which starts to come up in the fibrotic form of the disease. So I guess that's the best answer I can give.
Thank you. Wait short, please.
Thank you very much for the very nice presentation. I'm from Paris, and I'd like to ask a specific question about this of your study.
Could you hear me? There are a specific question about the piece of the study you demonstrated here. There is autoantibodies to several antigens that you found quite before the onset of the disease. So what do you think, when actually there is a starting point for the development of these antibodies, in this particular case for thyroiditis? Do you think that it starts earlier, their age could be during pregnancy, or it's later? And in connection with this, do you think that some of the autoimmunity may be more catastrophic, that may occur just before the onset of disease, and other kind of autoimmunity could be quite predicted a long time before the disease development? What is your opinion? Thank you.
Thank you very much. That's a very good question. I can tell you what I think, and perhaps why I think it, but it's not definitive, and that is I think that much of the antigen-specific signal is probably delivered long before the disease, and probably in many diseases in childhood. And perhaps epidemiologically, the best evidence is multiple sclerosis, where it seems that the stage is set for susceptibility before puberty, before the age of 12. And people who were raised in high endemic areas tend to remain high epidemic, even if they go to low epidemic areas. So that's the kind of study, and that's been true for several other kinds of autoimmune disease. So it may be that some exposure, let's say an infection, would be the logical one, not the only one, but certainly the most likely one, would be some infection in childhood. And interestingly enough, it would appear not a single kind of infection, but multiple infections. But then there may have to be a second signal. And that could be the same stimulus, the same infection coming back. But the experimental evidence suggests that it also could be another signal. And perhaps the best example of that is experiments that Fujiyama has done, in which he can sensitize rabbits with myelin basic protein very early, and then go back and give another antigen with point adjuvant at a much later time and exacerbate the disease. So that makes it more difficult to understand how molecular mimicry can work. It's obviously not a direct mimic, It's not that you have to have exactly the same epitope twice. What you need to have is a reactivation of what must be a relatively small proportion of memory T cells. And we still, at least I still don't have a clear understanding of what memory T cells actually recognize. But I think that's where the secret is going to come out. We're beginning to see, again, I'll refer to the EAE, multiple sclerosis model, that they may recognize more than the original antigen. They may recognize a different antigen. Referring to work of Government on this, which you may be familiar with, would suggest that they could recognize, that these prepared memory T cells could recognize a different, even apparently unrelated antigen. And I think that's intriguing, and it may be the heart of an explanation of why the second stimulus, the one that is really putting the train off the tracks, because the first one never does, as far as we know. The second one is the one that puts the train off the tracks. That apparently doesn't have to be the same infection or the same epitope that got the process started maybe at the age of 2 or 3 years old. So that's about as wild a speculation as I can provide at the moment. But it looks very much as if that's the way the train may be headed.
Thank you very much. Господа, давайте пожалеем доктора Роуза, который уже целый час стоит, дадим ему возможность присесть. И я хочу заметить, что сегодня и завтра на круглых столах мы будем обсуждать, там будет больше времени для высказываний, дискуссий о том, по каким признакам мы судим о возможности начинающейся болезни, когда нам вмешиваться. И самое главное, вопрос пациент, доктор, что у меня первый вопрос, второй, доктор, а что делать? Вот все это мы будем обсуждать, что делать. У меня тоже есть масса вопросов, я все-таки хотел бы, чтобы Роуз сел. Спасибо.